Fungi can reproduce asexually too. They can clone themselves. Sometimes we call it vegetatively, which is a bit of a misnomer because they're not vegetables, right? They're fungi. So they're completely composed of mycelium. And these fruiting bodies come in all sorts of shapes and sizes. This is an image from Biodiversity University, a BioU class that I teach here at the Nature Center every year. The next one's coming up in September. This is a collection of fungi that we grabbed right around the local area. And we've got coral fungi, we've got mushrooms that have teeth on the underside instead of gills. There's pored mushrooms that we commonly call boletes. A huge diversity of fruiting bodies. And they have super cool and interesting ecological roles. We have, anyone recognize these organisms? I have to say, the fruiting body is separate from the whole organism, right? So when you see a mushroom, there's a whole mycelia beneath the ground or in the wood growing. Here we have velvet shank, Amanita flaviconia, I forget the common name of that one. And then the one all the way on the right, I'll give you $10 if you can pronounce that right now. That is a parasite of an underground truffle, Tylopocladium ophioglossoides. This, here's the fruiting body, growing out of an underground truffle of the Elaphomyces granulatus. This all grows here. This is all from around here. So we've got decomposers, mycorrhizal fungi, and parasites. Those are basically the ecological roles of fungi. So decomposers, we call them saprotrophs. Here we've got beautiful oyster mushrooms. You've probably seen those. They're responsible for disassembling coarse woody debris. A vast amount of carbon is stored in wood, and fungi are fully equipped to access that food source. Their hyphae grow into the wood. They actually create enzymes based on what their hyphal tips are sensing in the substrate. There's an organelle that reads the landscape and says, oh, we need to make X, Y, and Z to decompose this. We need to make X, Y, and Z to set up a wall and protect ourselves from that fungus over there. It's really complex and interesting. A lot of our really familiar, oh, yeah, go ahead.
Sorry, are we holding questions to the end, or is it okay if I ask a quick question?
Yeah, hop in.
How long does that take? Because the building the wall or the sensing the ground, like when we talk, are we talking about evolutionary terms, or are we talking about like hours?
Hours.
Okay.
Hours or days. Yeah, there's some really interesting studies on oyster mushrooms, the mycelium growing through an agar plate with clean agar on one side, and then agar that has been polluted with petroleum. And it stops for like a couple of days and reads the landscape, develops the enzyme necessary to grow through the rest of the plate and consume the agar. Really neat stuff. So, to that end, these organisms in particular are really good bioremediators. And they've been used to break down petroleum pollution, maybe sopped up with straw or wood substrates out of an oil spill, for instance, and then sending the mycelium through that to eat it all up and decompose it. Really crazy. There's fungi that eat glass. I know. I digress. And then, of course, we've got the lovely chicken of the woods, Laetiporus sulphurea. Anyone eat this before? Collect it yourself? No. Nah? This is the first mushroom I collected on my own. It's a really fun feeling to be able to find something, identify it correctly, eat it, and not die. It's a good thing. It feels really good. I recommend it. And then we've got the red-belted polypore. that beautiful character. You've probably seen that, another decomposer. Here's the fruiting body. I'm just going to pass this around. You all can check that out while I'm talking. And turkey tail, a well-recognizable mushroom, highly medicinal. And wine caps, the Stropharia genus. These out here in the Nature Center is mulch. For probably 6 years, they've been fruiting just on their own. I think last year was the first year they haven't. We need more mulch and we need more mycelium out there, I guess. But they're really easy to cultivate and they're delicious. So then there are parasites. And like I showed you with the Tylepocladium, that's parasitizing another mushroom. We see fungi that are parasitizing insects. Are you all familiar with the story of cordyceps and how they turn the cordyceps or whatever the entomopathogenic fungus is, controlling the minds of the insect to crawl up to something really high? And this is an arthropod, polydesmid millipedes that are local. If you shake them up in your hands, they smell like cherry Coca-Cola. That's their defense mechanism. It's like a cyano-based or like a cyanide-based compound. If you were to eat it, it would be really disgusting. And you'd spit it out, not want to eat one again. But I was seeing these for years, like collecting on top of sticks like this. And I studied mycology in college and the early 2000s. And I kept thinking about the entomopathogenic fungi and thinking, I wonder if that's part of this story. Why do I find these organisms clumped together and dead? And basically, that is the story. A spore from the Arthrophaga genus attaches itself to the exoskeleton of this arthropod and begins to grow into the organism. And through a series of really interesting and probably new discoveries, folks are finding out how they do this, but it controls the insect or arthropod to climb up to something tall that has the perfect humidity and the perfect height so that this fungus can create a fruiting body. But what it does is it kills the organism, and they do like a death bite. They clamp onto something. It kills them, eats them from the inside out, creates a fruiting body, and then sends the spores out, and they do it again, and that cycle continues. There's some really interesting videos on ants. Just like go down a YouTube hole with zombie insects, You're gonna love it. Yeah, right? I think the show Last of Us is based on that. I haven't seen it yet, but I hear it's pretty interesting. But these parasites control and regulate populations, right? They're nasty. No one really likes a parasite, but they have a function. Like all of these organisms have a function in our world. And what people are learning is that they can use these organisms for population control of invasive pests. Something we're seeing now in the last maybe 5 years is an increase in the winter tick, which is decimating the moose population. Well, some folks have figured out that if you use Metarhizium, the genus for Metarhizium, it specifically attacks that species of tick. So they're coming up with these ideas of tossing out like these fungal bombs out into the forest to help keep the tick population down. Same with the spotted lanternfly. Ophiocordyceps is a genus that has been described as being useful for that. So there's ways to regulate a lot of these pests or invasive species, or species that we're seeing increase or move as the climate warms and changes, right? Which is why the winter tick population is so bad. Our parasites leave us with some delicious mushrooms. This is Hypomyces lactiflorum, which is a fungus that parasitizes Russula genus or Lactarius and create these really oddly misshapen fruiting bodies. And they're quite delicious. This is the lobster mushroom. Have you all had this? Cool. And so here, this is an organism that's a really interesting hybrid of the two ecological roles of a parasite and a decomposer. This is an Armillaria genus. A particular individual was discovered in the, I think it was the late 90s, to be inhabiting 4 square miles of forest and conservatively aged between 2,500 to 8,650 years old. So that's a wide margin of error, right? But it's about the biomass of a blue whale. This thing is really old and growing over a huge, vast portion of the earth. And what allows it to do that is this structure right over here. This is a bundle of mycelium inside a sheath of chitin, right? Chitinized sheath that is also melanized. What is melanin good for in an organism? Anyone know? Protection from UV radiation, yeah. So these organisms can grow these what are called rhizomorphs that grow into the ground, above ground, into trees. They parasitize trees and travel vast portions of the earth and parasitize the trees, then kill them, and then decompose their bodies. There's one individual can do that. So we've got an organism that is long-lived and well-established in forests. The honey mushroom, that's a common name for this organism.
Does it taste good?
Yes. They don't taste like honey, though. Yeah, they're like, Armillaria mellia is one that is, I think, is the honey mushroom, but you can call them all honeys. But it's like the honey color. It's like a bit of an amber. I was looking for the voice. Yeah, they're beautiful. Where I grew up, I think it was a Polish population. They called them polpinki. So I learned them as polpinki when I was a teen. We'd go out looking for them. They're pretty good. It's nice to know how to cook them. Every mushroom has its own style, right? But I recommend finding them. They come up in the autumn, late August, September around here, probably September around here. And then we have mycorrhizal fungi, which is the final ecological role I'm going to share with you. And essentially, it's a mutualistic relationship between fungi and plants. It is a relationship that's at least 95% of all vascular plants have it. I mean, it could be like up to 97, 98%. And it's a really complex suite of resource exchange between the plants and the fungi. So this is between kingdoms. And how it works is that the mycelium of a fungus infects or penetrates the root systems of, let's say, this oak tree. And in between the cells or into the cells, there's a few different varieties of how the mycorrhizal fungi infect the roots. But there's an exchange of nutrients between the tree and the fungus. The fungus reaches out into a much larger surface area to take up water for the plants. So it keeps the plants from fully desiccating or helps them be drought resistant. And it's reaching out into the substrate, accessing nutrients that the trees can't physically get to, as well as accessing nutrients that the trees have difficulty getting, like nitrogen and phosphorus. So the fungi are taking in these minerals and nutrients and shuttling that to the tree. And we could say in return, it's kind of complex, it's not totally like trading, but in return, the fungus receives sugars. What's interesting about this is the fungus, as soon as it gets the sugars from the trees, it reformulates it so it can't go back. Kind of like keeps it. But what's really interesting, there's some research indicating that plants have some degree of control about how nutrients flow through the mycorrhizal network. Because let's say we've got that bolete there. Let's say that bolete is growing into that oak tree. There's probably other trees that it's attached to as well, including relatives of that tree. And some folks have said that those trees can dictate where the nutrients go. When I learned about mycorrhizae in the early 2000s, we were under the understanding that the fungi were in control of where the nutrients were being shuttled. That's kind of breaking down right now based on people's research. So it's a little more interesting. And there's some really great research indicating that the fungi can hold on to resources and then shuttle them to the trees that are in need, like a tree that's ill or injured, that sort of thing, to keep that entire network of trees healthy. It's pretty complex. It's really cool. Of course, we've got the king bolete, delicious, the porcini. That is a mycorrhizal organism. We've got Caesar's mushroom. I think this is the North American species, Amanita jacksonii. And we have chanterelles. There's a few varieties here. We've got smooth chanterelles here. Black trumpets up there on the right. And then a wonderful bounty from a walk. Given all that we know about fungi, would you believe me if I told you that we have only described 6.5% to 7% of fungi on Earth? We don't know anything. It's amazing. There's a lot to be understood here. This is a view of Camel's Hump, I think from the north. So we're going to be talking about the fungi of Vermont from here on out and the conservation efforts that we're putting forth in the state. Before we go there, I just kind of want to set the stage for how these organisms interact with each other. Because fungi have been left out of the story for a long time. We have ram's head orchid. Orchids have an obligate relationship with mycorrhizal fungi. The orchid seeds lack an endosperm, and the endosperm is the initial energy package to sprout a seed. So they don't have that. So all the orchids need a mycorrhizal counterpart to infect the seed, to reach out into the substrate, supply the orchid seed with the nutrients to get started, right? Over there we've got a Russulus species. The Russulaceae have been known to be orchid mycorrhizae. Both of these grow here. I think these, anyone know their plants well here in Vermont? I think this is here, but it's rare or threatened, endangered. I'm not positive, I don't know. But that's the ram's head orchid. And then we've got, who's that in the middle? You know that one? The noisy little squirrel? Yeah, red squirrel. So, has anyone found mushrooms up in the trees in the autumn? That's from the red squirrels. So the red squirrels collect mushrooms, boletes. They love russula. They take them up into the tree, generally about like eye level for me, not too high up, just kind of like right about here. They stick the mushroom right on the branches, and they let it air dry in the autumn sun, in the autumn wind. And then when it's dried, they collect it and they stash it in their winter food stash. So they collect this for winter food. So this is happening with fungi. If you took away the fungus from this equation, the squirrel, I don't know, maybe the squirrel would be fine. They would find other sources of food. They'd work harder perhaps, or maybe move to a location where there's more mast species of seeds. But the orchid might not persist, right? They may not reproduce. So fungi are quite important. So about, I think it was 2022, I was invited to be a part of a group that formed at the state level called the Vermont Fungal Scientific Advisory Group. And we've got like a leadership team, and then we've got like general public members. So if you're really into this stuff, come see me. We'll get you in on the group. But basically our goal is to help inform the state on how to work with fungi, basically. But to get there, we have to understand, number one, what fungi are here in the state. We want to educate people on the value of fungal diversity and the importance of fungal habitat, how it all works together. And then we want to identify what species are rare, endangered, or threatened. Isn't that a beautiful coral? Super cool. To get there, we need to develop a comprehensive understanding of the macrofungi, meaning the fungi that we can hold, the fruiting bodies that we can pick up. Because there's a ton of fungi that we need microscopes to see or hand lenses to get down and check out, and they're super hard to ID, at least just with visual features. And then we're pointing towards the inclusion of fungi in environmental policy for ecological assessments, environmental health impacts, and conservation initiatives. If fungi is that important, it should be included in all of these things. We know that fungi function at an ecosystem level. So that's where we're headed and what we're working on. So this is kind of a tough nut to crack. We have to figure out how to collect all of this information with 10 people. You can't. So this whole thing is volunteer, by the way. This is just, we want to make this happen. So we came up with this idea of utilizing a really common platform called iNaturalist. How many of you have used iNaturalist before? Cool. How many of you heard of it? All right. So iNaturalist is essentially a crowdsourced digital database. And it works a little bit like this. See these 3 photos? These 3 photos offer features that could be used for identification of this fungus. Lacaria ochropurpurea. These are screenshots of my phone. You upload this app to your phone, and then you find a mushroom. Whether you know it or not, doesn't matter. You just walk up to it and say, wow, that's a cool mushroom. I'm going to take a picture of it and put it on iNaturalist. Maybe it'll help me ID it. So you walk up to it and you take good quality photos, the cap, the habitat photo, underside of the cap, how do the gills attach, maybe one with another mushroom standing by, some measurements, useful. You take some nice photos, and then you click on that little bit here that says, what did you see? And the software will generate some ideas for you. They're not 100% accurate, but it'll give you some ideas, and you can scroll through and say, well, I don't know if it's ochropurpurea or Lactaria subpurpureus or Haria chromipes. You could go down the list and say, I don't know how to do this. But you could say it looks like a Lacaria, and you can click on it, or you could just say, I don't know, and it gets dumped into the fungi category, into the kingdom. And then you save it, and you move on. The interesting thing is that it saves the location, which is important, the date and the time. You can set the settings to be private, like for instance, if it's in your backyard and you don't want people to know that there's this super cool mushroom that they could pick in your yard, like if it's an edible, or if it's really rare or unique. Or you could keep it open, which is what I do for most of my observations, unless they're like a highly sought after medicinal mushroom that I know someone's going to come clean the stump, that kind of thing. I'm like, I don't want that to happen. I'm going to hide this and just add that to my iNat page. So then you hit save and move on. To get, oh yes.
Okay. Here's my question.
Okay.
How do you avoid killing it? So you see a nice mushroom fruiting body and you want to to take pictures for iNaturalist.
Yeah.
You take a picture of the cap, and you want to get probably the gills, or whatever have you.
Uh-huh.
And doing this doesn't really work, so how do you avoid tipping the sucker over to get the picture that you need?
Super question. Well, this gets into ethics, right? Like, should we be picking all the mushrooms we find? Probably not. It depends on what it is, really. You can pick a lot of mushrooms and the organism will be fine. But sometimes you find one fruiting body in a whole forest of a unique fruit, a unique mushroom. What do you do with it, right? Well, if you don't want to pick it, you can get clever with your phone. You know how you do selfies like this? Send pictures to people? Just flip the camera around, put it underneath the cap, and take a photo. You know how that function works? That's one way to do it. Or you could pick it and tip it over and take a picture. I guess. I mean, there's not a great answer for it. Yes?
This one, the between open and private is obscured,
Yes.
and that's important because obscured is still, if you make it private, it's not useful for research purposes.
Yeah.
Obscured will keep it from people knowing exactly where it is, but it's still enough geographic information to know that it's in the county for research purposes.
Yeah, good point. Yeah, so there's different features of on how you make this private or obscured. I recommend going through the iNaturalist page, and you can see those designations and see how you want to upload photos. Thank you for pointing that out. I'll talk about that in a second. So if you want to know how to do this, we've got a page on the North Branch Nature Center website called the Fungi Vermont Program. And you can do the drop down in nature programs and go right to the page, and this will spell it out for you. And there's a few levels to this, right? So getting started, how to join. You don't have to join anything. You could just upload photos, but this is a really great resource to look in on. I recommend it. You can record good observations. That's step one, how to take good observations. Step number two is what we call better, which would be adding more notes, more details about the location, the fungus, what to measure, that sort of thing. And then step three would be, I take a picture of it, I record really good notes, upload it to iNaturalist, and I want to preserve the specimen as a voucher specimen so that we can do DNA analysis on it. So a lot of these fungi that we see in North America have relatives that look sometimes identical in Europe, or sometimes close enough that we don't readily see the difference. And with genetic analysis, genetic barcoding, we're finding that there's way more species here than we expected. Like, for instance, Amanitas. Like what we expected, one species to be has been broken out into 3 or 4 different species and continuing. And people are we're finding more species here that we didn't expect. New specimens, new species altogether even. So this is a great resource to drop in on. This is always open, you can check it out. So what happens with these observations? Like the ultimate goal is we're collecting information as a community. And by the way, you don't have to do anything special for it. You could go out with your kids and take pictures of mushrooms and boom, there you go. You don't even have to know who they are. I'll get to that in a second. But in 3 years, we've got 97,000 observations. That was like a couple days ago, I took a screenshot of this page. Super cool. That's a ton of information. Out of those, 1,697 organisms have gotten to a species. What we have here is what's called a project in iNaturalist, the Fungi Vermont project. Meg Madden created this project, and since she started it, it's been scooping up all of the fungi that are recorded in the state. Whether or not you mean to put it in here, it scoops it up, which is great for us. What we end up with is like all these observations. those are all the species in the middle. But what I want to point out is some of these features here, all this yellow needs ID, and the green, which is 26,135 specimens, are at research grade. What research grade means is when you upload a photo of a mushroom, let's say that Leucaria, I put it up there, I'm pretty confident I know what it is, and I label it as such. Someone, anyone, can find it and say, oh yeah, I think that's Leucaria too. And they'll click and say, I agree. And then if a third person says it's that species, it gets dumped into the research grade bin. That's what we're looking at, the scientific advisory group and other mycologists. We're looking at what has been identified 3 times by someone. Is it fully accurate? Not at all. People make mistakes and they miss things. So out of all those research grade observations, there's a lot that are not true to that species. So we're going back and trying to suss those out and figure those out. You all can do this. You all can go in iNaturalist, go to the Fungi Vermont project page, and just start sorting through pictures. Say, oh, I know what a chicken of the woods looks like. That's this, that's not it, and you can kind of go through it and help us out. So the benefits of collecting all of this information, number one, not one person has to do it all, or a team of paid scientists. We're collectively doing this. It's crowdsourced, and anyone can participate. Again, you don't have to know how to identify these organisms. It's a large sample size, which is useful. Large sample sizes are good. The identification is also crowdsourced. And observations can be used for further study. For instance, Amanita jacksonii. People are wondering how many are here in the state. Well, you can look and see how many observations have been uploaded to iNaturalist. Or you can focus on habitat types. You can really get a lot of good information out of this. That brings us to a really cool new update. The fungal SAG, as we call it, had acquired some funding to employ Vermont Center for Eco Studies to build a Vermont Fungi Atlas. And this is on their website. I think it's under Wilderness Projects, part of the Vermont Atlas of Life. And you can go to this and just search for species. If you look all the way to the right, it says 144 data sets. iNaturalist is one of those. So this is, iNaturalist is scooping a lot of information from other sources, including Mushroom Observer, which is what mycologists generally use to collect information digitally. We figured iNaturalist was so easy to use and people were using it already. Why try and turn everyone into a mycologist to use Mushroom Observer when we can use iNaturalist to get us to a good basal start? And then there's all the records on the left, all the contributors, images. If you go looking, you can just kind of poke around in here and just wander around. But you can also type in who you want to look at. And Amanita jacksonii, you can get some great info on this. For instance, pictures. Pictures are useful. How many records have been in Vermont? We've got 195. The first record, 2011, that's the first digital record. They've been observed here long before that. Last digital record, look at that, last year, August. And you can also scroll through that phenology calendar, and it gives you how many were found in this week, what the frequency was, that sort of thing. I think that says 43 out of 195 observations that year were in the month of July. So you can get some really great information. If you scroll around and look about, you'll find that there's a lot of information on taxonomy, Who are they related to? What is the family? That sort of thing. Also, information on how to identify them. Measurements, habitat type, etc., etc. Now I want to come back to this here. Do you see those S rank NA, IUCN NA, VT list NA? We don't have the information on those yet. But these would be like species of concern, threatened, endangered, that sort of thing. We're right now collecting the information to understand how we can designate these organisms. So that's on its way. So getting involved. I like to think, we can just all follow our curiosity, right? If you're into birds, explore birds. If you're into fungi now, explore fungi, right? Just be curious, ask questions. Interesting, I never saw this before. Let me take a picture of it. How can you contribute to this initiative? Well, you can get using iNaturalist. iNaturalist is a really great tool. I had a hard time getting to the point where I was happy taking photos in the woods all the time. So sometimes, I'll tell you, I leave my phone home, and those are when I find the really cool specimens. But I also think it's important to leave the digital stuff behind and to just enjoy nature. But do set up an iNaturalist account. You can use it on your phone. You can also take photos and upload them at home on a desktop too. You don't have to be carrying your phone around. You could use a digital camera if anyone uses those anymore. You can confirm IDs on iNaturalist to bring the observations we have to research grade or help work out some of that identification. And that's actually a really great way to learn fungi, is to look through iNaturalist and see how people are identifying these things. It's pretty useful. It's pretty neat. You can attend mushroom forays and classes. I highly recommend that. In-person learning is really important to get details of macrofungi that you can't really get in a digital format. You can also ask questions. You can talk about it. There's also something about learning a skill like this in a community of people, that you can ask them and say, hey, is this right? Am I seeing this right? I don't understand how these gills connect. Can you show me? It's so much fun to just do that together. And we're all looking for a way to be social too, honestly. We're all looking to connect. It's a great way to do it. And share your interests and passion for fungi with other people. Like I said, I work a lot with children, and I spend a lot of time talking with kids about fungi. They know how to be safe around them, and I'm happy to talk with people afterwards about this, how I work with kids in a safe way, and to foster that inspiration, that desire to learn more about fungi. And they know what to do around them. They know what's safe, what they can explore. And then, those kids that are 6 looking at mushrooms, where are they going to be in 20 years? I know kids that are 7 years old that know species names like that. It's so cool. So I want to share with you some learning opportunities in Vermont. And this is super cool. When I moved to Vermont about 7 or 8 years ago, I didn't hear anyone teaching classes. I didn't see anything really happening. And now there's a ton happening. It's so cool. Just last year, the Vermont Mycological Society began in Marlborough, Vermont, vermontmyco.org. We have 2 of their members on our leadership team now, and they're running forays at least once a month. There might be a couple of forays every month from May to October. And forays are basically meetings where you go out and you collect mushrooms, you take pictures of observations, you bring them back, dehydrate what is useful for collection, eat what is useful for food, and you can learn a lot in person that way. North American Mycological Society, or Association, NAMA, is offering their annual foray September 11th to the 14th, right down there in Marlborough, in that general area. Might be Plymouth. That's going to be super cool. If you really want to get your hands on some mushrooms and talk to some really interesting people and learn a lot, that's the place. They have walks you can go on. You can help with ID. You can really get involved. Northeast Mycological Federation has a foray the 18th to the 21st of September in Ithaca, New York. That's going to be super. That's me on Dr. Adventures. I run classes through that organization as well as through the North Branch Nature Center. If you're looking for me, I put this together, I was like, oh my God, this is giving me a panic attack. That's me and Mushroom. So on Dr. Adventures, that's my contact info. North Branch Nature Center, you can find me here during the school year. During the summer months, I am running just on Dr. Adventures courses. But the green classes are North Branch Nature Center. The maroon classes are on Dr. Adventures. Up next, May 10th, a mushroom walk here at the Nature Center. I hear it might be full, but I'm going to look at opening up the enrollment because I think we can probably get a bunch more people in. So if it's full, sign up on the waiting list. And that will be a walk for looking for spring mushrooms. I'm not sure how much is out, but we're going to look around and have some fun. Wild Mushroom Immersion, June 28th to October 18th. That's one Saturday a month for the fruiting season, basically. And that is for people who really want to dive in, learn about fungi, learn about conservation, learn about mushrooms as food and medicine. That's like kind of like learning in a community over the course of a season. I think it's the 12th year I'm running that, and it's a lot of fun. Mushrooms of the Northeast in Cherry Valley, New York, at Hawk Circle Wilderness Education Retreat Center. Wild mushrooms in cultivation, 2.5 days. And then here at the Nature Center in September, BioU mushrooms, field ID, and fungal ecology. And that, I think, is year 6 or 7 here. So there's some options to get learning, to get involved. And the idea, ultimately, is to include fungi in our long-ranging view of ecological systems and how we interact with the natural world. We know flora, we know fauna, but fungi are the organisms that are kind of like tying this whole globe together. Yeah, any questions? That's it. Okay, yes. Oh, thank you. Yeah, that'd be great. Thanks, Emily. Yes.
I have 2, and I don't know anything about mushrooms.
Cool.
These are like, they may be really dumb questions.
Not at all.
The first one is, if I'm going to take pictures of a mushroom, I'm very hesitant to touch it because I don't know whether I'm going to get something on my hands that later gets in my mouth and then I get sick.
Yeah.
So I feel like I need to stay like 10 feet away from these mushrooms. But
That's
anyway, so that's my first question. My second question is, are there fungi underwater, like in
Yeah.
lakes and ponds and
Yes.
oceans?
okay. Second question, yes.
Okay.
They're like, there's a lot of fungi. It's been a while since I've thought about them. It's been a couple years, but there's in fact there's fungi called chytrids, which is a group of fungi that cause a chytridiomycosis. They're parasites of amphibians, and they live in the water. They infect the keratinized parts of the amphibians, like around the mouth and around the sit patch where they drink water from. And they cause chytridiomycosis, which doesn't allow them to properly function. They get these weird outgrowths, and it's killing a lot of amphibians globally. In particular in the jungles, like the Dendrobatidae frogs, the poison dart frogs, their populations are really diminished because of that. aquatic fungus.
Right, oh, interesting, I'll post that up.
Yeah. As far as touching mushrooms, it's really great you asked that question. The only way you can poison yourself is by ingesting mushroom parts. So you could hold the destroying angel. You could crush it up and take a nap with it. Don't do that. But what I'm saying is you could handle them as long as you don't ingest mushroom parts into your body.
What if you like, you pick it, you know, there's lots around, so you feel like it's okay to pick it,
Uh-huh.
so you can take a photo, but now you've touched some of the liquid or the goo or whatever.
Yeah. Yeah. It's fine. There's nothing that's going to come out on your hands that is going to poison you.
Okay.
The only way you'll poison yourself is if you take a piece of that mushroom and eat it and swallow it. It's a really good thing to know. So, in fact, working with children, I set up a little wider boundary. Like, I tell them, don't touch any mushrooms until you know that it's not poisonous, right? Just a little safety in there. And that's especially important for really young kids. So number one poisonings are toddlers and puppies. Puppies and toddlers are the individuals who get poisoned. So something about mushrooms, we want to eat them, right? I don't know. So I tell the kids, don't touch the mushrooms. We can look at them. We can listen to them. We can draw pictures of them. We can talk about them. We can sit with them. But until an expert or someone that knows how to ID mushrooms sees it and says, this is a mushroom you can touch, it's safe to touch, I don't have them touch it. Now, there's a lot of fungi that are really cool to touch, like jelly fungi, the ones that are like jiggly, especially jelly tongue, the one that looks like a tongue emerging from a log that jiggles, pseudohydnum, yeah. So they're worth exploring. They're tactily interesting, but you can't poison yourself by picking one, tipping it over. You know, extra safety, if you're worried about it, you can always wash your hands afterwards, right? To just make sure there's no bits and pieces under your nails or anything. But generally, that's not really enough to do any damage. But mushroom poisonings are serious. I would check out the North American Mycological Association's webpage and look up the poisonings. They have a whole registry of poisonings. You can find out what people have eaten and poisoned themselves with, how it happened. You can find out how the experts poison themselves, which is interesting. Doesn't happen much, but it happens, you know. Good question. Yes?
So this is when it comes to foraging. At first I heard that, like with morels, for example, that you shouldn't, if you find a patch, don't take them all. Just take maybe about a third of them and leave the rest. They need to spore. And then I heard that, well, as long as you harvest and using a net bag so the spores can fall out as you're going along, then it's okay to harvest them all because it's more like picking apples off a tree. You're not killing the organism, just taking the fruit.
Yeah.
So, is there a way for you to, when you're foraging, can you tell by observation if the mushroom has already let its spores go?
Sometimes you can tell if they're mushrooms that grow together in a group, like a bouquet. They'll be stacked on top of each other. And sometimes you can see the spores landing on the other cap.
Okay.
Or you can see the spores have landed on the stem. Like it'll be like a dusting of color. So I know both of those ideas about, you know, the ethics of collecting, basically. People have mixed opinions about it, including mycologists. There's been some studies that have shown that, like, yeah, you can pick all the chanterelles in the forest, it's fine. But I question that, honestly. You know, I think there's something to be said for thinking about other organisms, not only the fungus, but all the other organisms that might depend on the fruiting bodies, right? Like I mentioned, the squirrels collecting all those recula. So there's other organisms at play here too. So, you know, it depends on the organism, really. And it's true having a basket or a mesh bag can help spread those spores, kind of like Johnny Appleseed type character. You're walking through the woods with your basket, spreading spores everywhere. That's a good thing. But as far as like a definitive answer, you know, I tend not to collect more than I need. So I decide what my need is. I don't sell mushrooms to restaurants. It's just something I've chosen not to do. So when I collect, it's not for money. It's for my own personal use and for medicinal use, or if there's a class, we're collecting for the class. So I think about the organism first. They're creating fruity bodies. They must be necessary, right? So I will collect maybe 50% or 25%. It really depends on what it is. But if you find one morel, do you eat it? I'd probably pick it and eat it. You know, the other thing is like there's, you know, you can kind of lean on this. There's probably more around that you haven't seen yet, or they're going to pop up. There's been some interesting studies that show like, you know, fruiting will sometimes propagate more fruiting too. And you can actually force mushrooms to fruit in cultivation that way. Like you collect them, then you kind of starve them and dehydrate them and then water them and then they fruit again. There's some really interesting things with fungi. So they're not quite like plants. They don't function like plants. Chances are the organism's okay, but there are fruiting bodies that people haven't seen before from a species. There's been some interesting studies where some folks like set up an acre plot and they followed it. They followed the fruiting for like 20 years and they're still finding new specimens in that little bit. So it's a complex answer to a complex question, I guess. Yeah, but use your best judgment. What feels right? Yeah. Okay, yes?
So we cultivate medicinal mushrooms at home that we forage, or we cultivate and we also forage.
Cool.
So this is like our first winter doing that here in Vermont. So I've spent my time in the winter with some chaga, with our birch polypore,
Mm-hmm.
whatever. I'm like becoming like a major stalker, like waiting for some new turkey to come out or whatever. When do we start seeing some sort of new signs of life? Like we own 10 acres by Northfield and
Okay.
I'm becoming like a creeper. Like I've got a couple of logs that have some old white turkey on it and I'm just touching them like
Uh-huh.
I just want to like start again.
Yeah.
And is the white like older hard turkey, like does that still have medicinal properties? I'm gonna feel like no, but
Yeah.
maybe two questions on it.
Yeah, you want to collect fruiting bodies that are fresh so that those like constituents haven't broken down through the course of winter and decomposition. Turkey tails, I think, if I'm not mistaken, I think you'd probably start seeing them fresh in August, end of July. What you can also do is go on to the Vermont Center for Eco Studies, go onto that website, look up turkey tails and see when the frequency of fruiting in Vermont is. So you could just go right there and see how many people are seeing that specimen and when and where even. You can find out where.
But just like in general, like is it closer to June, July that we
For
start
turkey
seeing-
tails
No,
or
just
in general?
In general.
Oh,
just in
okay.
general.
In general, things really start to kick off end of June, beginning of July. Like end of June is when I generally start teaching mushroom classes, and because there's a greater diversity. But there are fungi that are beginning to fruit right now. It's just the diversity is lower. And then end of June starts to kick off. July is really good. August really good. September. In October, it begins to like start decrease in frequency. But we'll get mushrooms like in through November and December too. Yeah. Yes.
So, are there any mushrooms that we should worry about introducing? Like you mentioned the wine caps.
Yeah.
And so like I, put some wine cap spore in our wood piles and then it started to pop up in the woods.
Yeah.
And I haven't seen it anywhere else. So like, is there, with all the varieties that are like available as spawn, should we be thinking about invasive species of fungi?
Yeah. It's a super question. I don't know a lot about the competition between fungi that are introduced and not. But there is competition. About 5 or 6 years ago, I was in New York State, west of the Hudson River near Albany, New York, teaching a mushroom class, and I found, excuse me, yellow oyster mushrooms propagating the forest. They're a European species. I think they're from Russia, that area. But they're cultivated. And I looked at the occurrences, and from Ohio, they are moving east. Excuse me. So they're causing a bit of a problem for the local native oysters. So it is a problem. I don't know if anyone's really gotten a handle on it yet. I think you could probably take one species and study that and see how it's affecting everybody. But it is a concern. As far as wine caps, I don't know their natural history. I know there's native Stropharia, but I'm not sure about those. Excuse me. Yeah. I would do a little more looking into it. I don't have great answers for you there, but there is an impact. Actually, one thing I can say, when you're buying a mycorrhizal dip for your plants, make sure they're native mycorrhizae. That's something we're seeing where there's just like a host of mycorrhizae that people are putting in this powder that you put on the end of your roots, and then you're introducing a non-native species to the soil structure. I know that's something that's a bit of a problem now. Yeah, super. Okay, yeah.
Hi.
Hey.
I love taking photographs of mushrooms. I don't know how to identify them, and I've never foraged. So I have two questions. One is, when you forage for mushrooms, are you only allowed to forage on your own property, or do people forage on state land or anywhere? So that's one question. And the other is, I think you mentioned that you could take photos that stored on your computer and upload them to iNaturalist, and I know where I've taken them and when, but it's not going to have the GPS information. Is that still okay?
Yeah, you'd be surprised. It depends on how you're taking the photos. If you're taking them with your phone and you haven't changed any of the settings, chances are the GPS location and the time and the date is nested in with the photo. And when you upload it, it all gets sucked into iNat.
Yeah, well, some I've taken with a digital camera.
Okay. Yep. So those, knowing that it's in Vermont alone is helpful. Knowing the exact location is better, but if you don't have that, you can still upload them. And I think,
Type in the town name.
yeah, I think you can type in the location.
I do that a lot with my camera, and it pops up a little map, and there's a blue dot, and you can just take it to where
Oh, yeah,
you took
yep.
the picture.
Yep.
So I take a lot of photography in Florida, and then I come home and I'm sorting through my pictures, and I'm like, oh, I didn't post that one, but I know where it was.
Mhm.
So
Yep.
you can do it after the fact.
Cool. Good. Great question. Did I answer them?
And then, yeah,
Was there another one?
thank you, but then there's the one about where you're allowed to forage.
Oh, foraging, right. Check on the state's websites. Of course, private land, is the best. I'm pretty sure that on state land, it's okay to forage. Generally, most towns don't have, in Vermont at least, don't have any sort of prohibition on that, unless you're collecting for sale. And that's where I think it changes. So generally what I'll do, like for instance for 4As, or if I'm bringing a class somewhere, I'll contact the state and say, hey, I'm going to bring a group of people to this area. Can I have permission to collect for scientific purposes, that kind of thing? And they're generally really great about it. So collecting for yourself on state land, I think, is just fine for food. But it would be worth double checking. Yeah. Yeah, check with the state's website. Yes.
More of a comment, going back to the question about touching mushrooms. My mother tells me that my Polish grandmother who immigrated here used to touch the mushroom on her tongue to help identify whether or not it was edible or not.
Yeah.
And she lived to a ripe old age.
And she made it. Right. That's like a testament to you have to eat it, right? To
Right.
poison yourself. So I'm glad you brought that up. If you look through some of the field guides even, you open up a field guide and it'll give you all the details of the fungus, the fruiting body, and then it'll say it tastes acrid. And you're like, oh man, I gotta taste this thing? Well, I don't recommend tasting something unless you can get it to the genus and you're confident like it's right about there. In which case, like, don't do this for a little bit. You need to learn some skills first. But the idea is there are fungi, and you hear this a lot from, immigrants that know that this group of, for instance, these group of boletes, there's ones that you can nibble on and they're acrid or bitter. You don't want to eat those, so you spit it out and leave them. But then there's some that look almost identical that if you put them in your mouth, they taste good. And that's like another identifying characteristic that people have used. So a lot of times when people come to North America, they poison themselves because they're different species, but they look pretty similar. So they're like, oh, cool, this is the mushroom I grew up eating. And if you think about people in their local bioregions, like let's say Poland or, I don't know, Slovenia, something like that, they're in a village and their grandparents brought them out into the hills to the same places, and they know that there's 3 or 4 species, or maybe 2 species that look similar. And one of the ID characteristics is one tastes bad and one tastes good. And you eat the one that tastes good. So like, it's not necessarily the best diagnostic, but it works in some conditions. But there is something to tasting a mushroom and then spitting it out and rinsing your mouth. Again, I recommend being in the presence of someone who's done that can tell you this is okay to put in your mouth and taste.
Right.
And this one, if you accidentally swallow it, you'll die. You don't want to be in that situation. Good question or comment. Yeah, cool. Awesome. Well, if anyone wants to keep in touch with me through the summer, I've got an email sign up. Think of donating to the Nature Center to keep programs like this moving. We do a lot for the community and a lot of subsidies for programs, including school-age programs through the ECO program. A lot is offered through donation. So do consider donating. And thank you all for being here.